this annual organization here now. Today we shall take up the remaining concepts here now. We have done till skeletal muscle in the animal organization here now. In today's class we shall talk about the remaining muscular tissues and neuron. Now the skeletal muscle you learnt already here now. Then we will start with the visceral muscle here now. Visceral muscles are also called smooth muscles. Visceral muscles, they are called smooth muscles, and most of the visceral organs are made up of these muscles. That's why they are known as visceral muscles. Except heart, all the internal organs in human body are made up of these visceral muscles here now. They are also called smooth muscles, and they are non-striated and involuntary. They are non-striated and involuntary. Non-striated and involuntary. Now these muscles, they occur in the form of sheets or bundles. They occur in the form of thin sheets or bundles here now. Now each muscle fiber is spindle shaped, you know. Each muscle fiber is spindle shaped with tapered ends. In skeletal muscle, each muscle fiber is cylindrical with blunt ends, whereas in smooth muscle fibers, each muscle fiber is spindle shaped with tapered ends. And it is uninucleated. You know. Unlike skeletal muscle, visceral muscle is uninucleated. That is, skeletal muscle is syncytial with many nuclei without cross walls, you know, whereas the visceral muscle is uninucleated and spindle shaped with tapered ends. The smooth muscle fibers are of two types here now. They are unitary smooth muscle fibers, multi-unitary smooth muscle fibers, now unitary smooth muscle fibers here now, the several muscles are arranged closer to each other. They are all spindle shaped with tapered ends. They are arranged closer to each other here now, and all of them contract collectively as a single unit here now. All of them contract collectively as a single unit. They are under the innervation of a single nerve fiber. Whereas multi-unitary smooth muscle fibers, the muscle fibers are spindle shaped with tapered ends, and they are independent of each other with regard to their contraction here now. They are independent of each other with regard to their contraction. Here each muscle fiber is under the control of a single nerve fiber here. Here all of them contract collectively. Here the contractions are independent of each other. Each muscle fiber is under the innervation of a single nerve fiber. So such smooth muscles are called multi-unitary smooth muscles. When all of them contract collectively as a single unit, we call them unitary smooth muscle fibers. You know, multi-unitary smooth muscle fibers are present in iris, ciliary body, piloerector muscles, and muscles of eyelids. Okay, iris, ciliary body, piloerector muscles and eyelid muscles are of multi-unitary type here now. Then all the other smooth muscles belong to unitary smooth muscle type here now. All other smooth muscles, like the muscles present in blood vessels, lymph vessels, digestive glands and their ducts, then alimentary canal from esophagus to the rectum, mid part of esophagus to rectum, then urinary bladder, then dermis of skin, okay, dermis of skin, then the respiratory tract, then uterus. So all these belong to unitary smooth muscle fibers. Blood vessels, lymph vessels, digestive glands and their ducts, alimentary canal, that is gut, from mid part of the esophagus to rectum, urinary bladder, dermis of skin, respiratory tract, okay, then uterus, all of them are of unitary smooth muscle type, whereas iris, ciliary body, pilorectal muscles and eyelid muscles are of multi-unitary smooth muscle type here now. Now here the both of them contractions are involuntary and they are non-fatigable here now. Okay, then they have got rich blood supply here now. They do not get fatigued upon stimulation here now. Contractions are slow and sustained, unlike that of skeletal muscle fibers here now. And the smooth muscle fibers here now, they have certain undifferentiated cells called pericytes. Undifferentiated cells called pericytes are present in the smooth muscle fibers, and these pericytes are responsible for regeneration of smooth muscle fibers here. Even in smooth muscle fibers also, thick and thin filaments are present, that is actin filaments and myosin filaments are present. But unlike the skeletal muscle fiber, the thick and thin filaments are scattered in the sarcoplasm here. In case of skeletal muscle, the thick filaments are confined to A band, whereas thin filaments are confined to I band here now. That confined organization is lacking in case of smooth muscle fiber. That's why in spite of the presence of thick and thin filaments here now, smooth muscle fibers appear non-striated here. Then the next one is the cardiac muscle fibers. The cardiac muscle fibers here now, they are involuntary and striated. They are present in myocardium of heart and base of the large blood vessels. The basal part of aorta and the myocardium of heart are made up of cardiac muscle fibers. The cardiac muscle fibers, each cardiac muscle fiber is again cylindrical, like that of skeletal muscle fiber with truncated ends. Okay, they are uninucleate or binucleate. Uninucleate or binucleate here now. Then the cardiac muscle fibers are arranged end to end like this. They are arranged end to end and they are all branched here now. Okay, all of them are branched. The only muscle fiber which is branched is the cardiac muscle fiber. Like this, these cardiac muscle fibers are branched now, and branches are connected to each other to form a network. Each muscle fiber is uninucleated or binucleated here now. Then at the point of joining here now, the plasma membrane, the sarcolemma, is thickened to form structures called intercalated discs. Intercalated discs. The presence of intercalated discs is one of the unique characteristic features of cardiac muscle fibers here now. The intercalated discs contain gap junctions and desmosomes. The desmosomes act as anchoring junctions between the cardiac muscle fibers, while the gap junctions act as ionic channels, that is, they help in conduction of ions and conduction of impulse from one muscle fiber to another muscle fiber here now. The gap junctions are also called nexuses. The gap junctions are also called nexuses. They help in conduction of impulses from one muscle fiber to another muscle fiber. They also facilitate diffusion of ions from one muscle fiber to another muscle fiber here now. Okay, due to the presence of gap junctions and branch network here now, cardiac muscle fibers, though they are uninucleated, functionally they act as a single unit here now. Functionally they act as a single unit. That is, upon stimulation, all the cardiac muscle fibers contract at once, acting as a single unit, Hence, cardiac muscle fibers are referred to as functional syncytium. Are referred to as functional syncytium. With regard to functioning, all the cardiac muscle fibers act as a single unit and they contract at once now, and that's why it is referred to as functional syncytium. That is, with regard to functioning, when you take, it appears as though it is multinucleated contracting as a single unit here now, hence it is referred to as functional syncytium. Within the cardiac muscle fibers, there are certain autorhythmic fibers here now, which are capable of eliciting contractions within the cardiac muscle fiber on their own. Those cardiomyocytes which exhibit autorhythmicity, collectively they form a pacemaker. Collectively they form a pacemaker, that is sinoatrial node. Sinoatrial node acts as a pacemaker. It lies in the right auricle at the base of inferior vena cava near Eustachian valve here now. So that sinoatrial node is responsible for systolic and diastolic movements of the heart when the heart is filled with blood here now. Okay, and that's why kind of contractions are called autorhythmic contractions. Autorhythmic contractions. Like smooth muscle fibers, the cardiac muscle fibers are also non-fatigable. They are also non-fatigable here now, and they are striated here, like skeletal muscle fibers, they are striated. That is, even in cardiac muscle fiber here now, thick filaments are confined to the A band, thin filaments or actin filaments are confined to I band here now. Because of that, the muscle fibers appear striated here now. And cardiac muscle fibers have rich blood supply here now. They have got abundant amount of ATP, myoglobin and phosphocreatine within the sarcoplasm. That's why the cardiac muscle fibers never fall short of ATP or glucose during contraction here now. This is one of the reasons for the cardiac muscles remaining non-fatigable during contraction here now. And refractory period for the cardiac muscles is also very short here now. As a result, the cardiac muscle fibers here now, okay, I repeat, the refractory period of cardiac muscle fibers is much longer than that of skeletal muscle fibers. As a result, the cardiac muscles do not undergo fatigue here now, because during refractory period they replenish the entire energy that is lost during contraction here now. Now we shall talk about the connective tissue, that is nerve tissue here now. Nerve tissue is basically made up of two types of cells here now, that is neurons and neuroglial cells here now. They exhibit the characteristics like irritability and excitability and conductivity here now. They exhibit characteristics like irritability, excitability and conductivity here now. Basically, the nerve tissue is made up of two types of cells, neuroglial cells and neurons here now. Neuron happens to be the longest cell in the human body here now. Neuron was discovered by Fossil and Hiss. It was discovered by Fossil and Hiss here now. Now neuron, it consists of a structure called cyton. Cyton is also called soma or cell body or perikaryon. Cyton is also called soma or cell body or perikaryon. Cyton is deposited with cytoplasm. The cytoplasm content of cyton is called neuroplasm. The cytoplasm content of cyton is called neuroplasm. Now within the neuroplasm, there are certain ribonucleoprotein depositions or particles. The particles which are made up of ribonucleoproteins are called Nissls granules, or they are also called tigroid bodies. Okay, the structures which are made up of ribonucleoproteins are called Nissel's granules or tigroid bodies. The tigroid bodies are present in cyton and dendrites, and they are supposed to synthesize the neurotransmitters which help in conduction of impulse from one neuron to another neuron. Apart from Nissel's granules, there are several neurofibrils within the cyton. There are several neurofibrils within the cyton. And in certain neurons, certain pigment granules called lipofuscin granules are also present. Lipofuscin granules are also present within the neuroplasm. The lipofuscin granules are supposed to be the metabolites that get accumulated in the neuroplasm, and they envisage the senescence of the neuron. That is, when the neurons grow older, they get deposited with the metabolic depositions called lipofuscin granules. Okay, then the cyton, which is also called soma or cell body or perikaryon, it gives rise to two kinds of fibers. The fibers that are given off from cyton are called dendrites. All these fibers are called dendrites. The dendrites are also called efferent fibers. The dendrites are also called efferent fibers. The dendrites or efferent fibers are made up of neuroplasm. They are also deposited with Nissel's granules and neurofibrils. They form synapse with the teledendrites of a preceding neuron. Now, the cyton gives rise to a long fiber called axon, which is also called efferent fiber, or it is also called nerve fiber. Efferent fiber or nerve fiber. The axon is deposited with the cytoplasm. The cytoplasm content of axon is called axoplasm. The axoplasm. The outer layer of axon is called axolemma. The outer layer of axon is called axolemma. The axon is slightly bulged at its proximal end. This antemost bulged part, the proximal bulged part of axon, is called axon hillock. The axon hillock gives support to cyton. Sometimes the axon gives rise to lateral branches called collateral branches. Then towards end, the cyton branches to form terminal arborization. Terminal arborization. The cyton branches to form, ramifies to form terminal arborization, or they are also called teledendrites. The teledendrites or terminal arborizations are swollen with synaptic buttons. They are swollen towards the tip to form synaptic buttons or synaptic knobs, you call them. The synaptic knobs of one neuron form synapse with the dendrites of another neuron. So, with regard to the basic structure of an axon. Now, many axons or nerve fibers come together to form a nerve. Many axons, like these are all axons many axons come together to form a nerve, and the nerve is covered by a connective tissue sheath called perineurium. The connective tissue sheath that ensheaths a nerve or a bundle of nerve fibers is called perineurium. And this is an axon. Axon is covered by a connective tissue sheath. It is covered by a connective tissue sheath. Each axon is covered by a connective tissue sheath called endoneurium. And a bundle of nerve fibers or nerve is also called fascicle. Okay, it is also called fascicle. So the connective tissue sheath that covers a fascicle is called perineurium. Many fascicles are held together by an outermost connective tissue sheath called epineurium. So remember, MCET and APMT point of view, the most important point many fascicles are held together by an outermost sheath called epineurium. Each fascicle, that is a bundle of neurons, is covered by perineurium, and each nerve fiber or axon is covered by endoneurium. The bundles of nerve fibers within the central nervous system, that is in brain and spinal cord, are called tracts. Bundles of nerve fibers within the central nervous system, that is brain and spinal cord, are called tracts. In the same way, the clusters of cytons, aggregation of several cytons within the central nervous system, are called nuclei. They are referred to as nuclei, and aggregations of cytons in the peripheral nervous system are called ganglions. If many cytons come together to form a solid deposition like structure, you call it ganglion in case of peripheral nervous system, and within the brain and spinal cord, those aggregations are collectively called nuclei. Now, we shall talk about the types of neurons on the basis of their structure. With regard to the structural classification here now, you may come across two kinds of neurons myelinated neurons and non-myelinated neurons Myelinated neurons are also called medullated neurons In myelinated neurons the axon is covered by a myelin sheath Axon is covered by myelin sheath. Like this, there is a deposition of myelin here around the axon, and that myelin sheath is, this is the myelin sheath here this is the myelin sheath, which is also called medullary sheath. Myelin sheath or medullary sheath here Myelin sheath is interrupted at certain places to form nodes of Ranvier. Nodes of Ranvier. Interrupt at certain places to form nodes of Ranvier. In peripheral nervous system, that is cranial nerves and spinal nerves myelin sheath is interrupted at certain places to form nodes of Ranvier, while within the brain and spinal cord myelin sheath is continuous without any nodes of Ranvier. Then myelin sheath, with regard to the peripheral nervous system, is secreted by spindle-shaped cells called Schwann cells. So these spindle-shaped cells which are present, one in each internode, they are called Schwann cells. Each Schwann cell secretes a myelin sheath in a concentric manner around the axon here Like there are several Schwann cells encircle themselves around the axon to form several concentric layers of myelin sheath until the axon attains considerable thickness Then once a myelin sheath is deposited around the axon to a larger extent then the Schwann cells remain intact to form a layer. The Schwann cells remain intact to form a layer around the axon. That layer of Schwann cells which lies around the axon is called neural lemma. The layer of Schwann cells around the myelinated axon is called neural lemma. Neural lemma is continuous even over the nodes of Ranvier here Due to presence of nodes of Ranvier, impulse, instead of traveling with the same velocity all through the neuron, it jumps from node to node. That is, action potentials created only at the nodes of Ranvier so that impulse can reach the target organ fast. That mode of conduction of impulse, where the impulse appears as though it jumps from place to place or node to node, is called saltatory conduction. However, at the node of Ranvier, impulse is prevented from straying into surrounding tissues by neural lemma here And remember, dear students, in case of peripheral nervous system, myelin sheath is secreted by Schwann cells, whereas in central nervous system like brain and spinal cord, myelin sheath is secreted by oligodendrocytes. So neurons with the myelin sheath are called myelinated or medullated neurons. Myelinated neurons are present in white matter of brain and spinal cord and cranial nerves and spinal nerves of peripheral nervous system. The neurons which do not have myelin sheath are called non-myelinated nerve fibers. In non-myelinated nerve fibers due to the absence of myelin sheath, they appear grey. That's why they form grey matter within the brain and spinal cord. So grey matter of brain and spinal cord and the sympathetic and parasympathetic system most of the neurons are non-myelinated. In non-myelinated neurons, the leakage of impulse is prevented by neural lemma. Myelin sheath is absent, but they are covered by neural lemma to prevent leakage of impulse from the axon With regard to structural classification, you come across two types, that is myelinated neurons and non-myelinated neurons. Then on the basis of the number of dendrites that arise from the cyton, we divide them into 3 more types, that is unipolar neurons, bipolar neurons and multipolar neurons. If a cyton gives rise to several dendrites like this, it is called multipolar neuron. Multipolar neurons are mostly present in cranial nerves and spinal nerves. If a cyton gives rise to a single dendrite which is highly branched and a single axon, on the basis of the structure, you can divide the neurons into 3 types. Multipolar neurons, bipolar neurons, unipolar neurons. Now multipolar neurons, the cyton gives rise to several dendrites. It gives rise to several dendrites and a single axon. Multipolar neurons are present in cranial nerves and spinal nerves. Bipolar neurons, the cyton gives rise to a single axon and a single dendrite which is highly branched. Cyton gives rise to a single dendrite which is highly branched and a single axon. Such neurons are called bipolar neurons. Bipolar neurons are present in cochlear and vestibular ganglions. They are present in cochlear and vestibular ganglions of internal ear. They are also present in olfactory neuroepithelium. Olfactory neuroepithelium of nasal chambers, and present in retina of eyeball. So in retina, olfactory neuroepithelium, cochlear and vestibular ganglions of internal ear, bipolar neurons are present here. Unipolar neurons, the cyton is laterally placed. It gives rise to a single fiber which acts as dendrite at one end and axon at the other end. Cyton is laterally placed. It gives rise to a single fiber which is differentiated into dendrite at one end and axon at the other end. Unipolar neurons are also referred to as pseudo-unipolar. Unipolar neurons are present in dorsal root ganglion of spinal cord. Pseudo-unipolar or unipolar neurons are present. And sometimes some neurons are without any dendrites, you call them apolar. Apolar neurons are present in neuroblasts of the embryonic stage. Now on the basis of mode of functioning, you can divide the neurons into about 4 types. First one, you have sensory neurons. Sensory neurons are also called efferent neurons. The neurons that carry impulses from the skin, then the receptors and somatic organs to either spinal cord or brain are called sensory neurons or efferent neurons. The next you have the second one, motor neurons or efferent neurons. The neurons which carry messages from brain or spinal cord to the effectors can be called motor neurons or efferent neurons. Then you have mixed neurons. If a neuron is made up of both sensory nerve fibers and motor nerve fibers, it is referred to as mixed neuron. That is, within a nerve, some nerve fibers act as sensory nerves and some of them act as motor nerves. Such neurons are called mixed nerves. The next you have another one, interneurons or association neurons. The neurons which are there within the brain and spinal cord are called association neurons or interneurons. So with regard to functioning, neurons are divided into 4 types: sensory or efferent neurons, motor or efferent neurons, mixed neurons, and the next one is interneurons or association neurons. Now as I made a mention earlier, the synapse is the proximity of dendrites of one neuron and teledendrites of another neuron. Supposing this is a dendrite here now with a synaptic knob. This dendrite with a synaptic knob lies near teledendrite of another neuron. This neuron is referred to as presynaptic neuron, and the other one is called postsynaptic neuron. And this is the presynaptic membrane. Okay, the presynaptic neuron has several synaptic vesicles. The synaptic vesicles store neurotransmitters. Synaptic vesicles are deposited with different kinds of neurotransmitters. Then within the presynaptic membrane, there are several calcium ligand channels are present here. Several calcium ligand channels. Are present. So at the time of conduction of impulse now, the calcium ions from the extracellular fluid diffuse into the synaptic knob here now through calcium ligand channels, and thereby bring about exocytosis of synaptic vesicles to discharge neurotransmitter into the synaptic cleft. Now the gap that lies between them is called synaptic cleft. And the postsynaptic membrane, this is the postsynaptic membrane here now. Has several ligand gated channels. Okay, it has several ligand gated channels. So when neurotransmitter interacts with the ligand gated channels of postsynaptic membrane, all the ligand gated channels of postsynaptic membrane get depolarized to facilitate conduction of impulse from presynaptic membrane to postsynaptic membrane here now. And the structure of synapse was studied by Sherrington. The structure of synapse was studied by Sherrington, and the mode of functioning of synapse was elucidated by Henry Dale. The mode of functioning of synapse was elucidated by Henry Dale, and structure was studied by Sherrington here now. Okay, so here remember, within the presynaptic membrane there are several calcium ligand channels now. So at the time of conduction of impulse from presynaptic neuron to postsynaptic membrane, the calcium ions from extracellular fluid diffuse into the synaptic knob through ligand gated channels of the calcium, and they bring about exocytosis of synaptic vesicles to discharge neurotransmitter. Neurotransmitter picks up the wave of depolarization from presynaptic membrane. It diffuses across the synaptic cleft and interacts with the ligand gated channels of postsynaptic membrane to elicit depolarization, wave of depolarization at the postsynaptic membrane. From the postsynaptic membrane, the depolarization is conveyed to cyton, and it travels further through axon to telodendrites of another neuron here now. So this entire structure was studied by Sherrington, and mode of functioning was put forth by Henry Dale here now. And neurotransmitters, they are broadly divided into two types here now: excitatory neurotransmitters and inhibitory neurotransmitters. Adrenaline, acetylcholine, then dopamine, then serotonin. Okay, they can act as either excitatory or inhibitory neurotransmitters, whereas glycine and GABA, GABA is gamma aminobutyric acid. Glycine and gamma aminobutyric acid, both of them always act as inhibitory neurotransmitters. Mostly the nerve terminals of sympathetic nervous system synthesize adrenaline or norepinephrine. That's why they are called adrenergic nerve fibers. For adrenaline, monoamine oxidase. For adrenaline, monoamine oxidase acts as a catalytic enzyme here now, and the nerve terminals of parasympathetic system mostly secrete acetylcholine here now. That's why they are referred to as cholinergic nerve terminals. For acetylcholine esterase acts as a catalytic enzyme here now. So remember, for acetylcholine esterase acts as a catalytic enzyme, whereas for adrenaline, monoamine oxidase acts as a catalytic enzyme here now. Now we shall talk about the animal organization too. The important characteristic features of various invertebrate phyla. They are also the most important concepts here now for your AFMC, JIPMAR, AIMS, APMT and MSET. We shall emphasize on the most important points which are likely to appear in the various competitive exams here now. Now first we shall start with under invertebrate phyla, we shall start with phylum Porifera. Important characteristic features of phylum Porifera. Now phylum Porifera, the term Porifera was coined by Robert E. Grant. The term was coined by Robert E. Grant. The term Porifera was coined by Robert E. Grant. Huxley and Solas separated them from metazoa and placed them under parazoa. Separated them from metazoa and placed them under parazoa. The term parazoa was coined by Solas. Okay, parazoans were created by Huxley and Solas, separated them from metazoa, and the term parazoa was coined by Solas. The study of poriferans is also called parazoology. Poriferans are referred to as the blind offshoot of evolutionary ladder. Blind offshoot of evolutionary ladder. Many reasons can be attributed to that characteristic feature. One of the most important reasons for calling them blind offshoot is they have evolved from the protorospongians of phylum Protozoa, but did not give rise to any higher phyla. Unlike all the other invertebrate phyla, they evolved from protorospongians of phylum Protozoa, but did not give rise to any other higher phyla. One of the main reasons calling them blind offshoot of evolutionary ladder.